Hello, everybody. Welcome back to our e-learning genomics workshop on genomics and bioinformatics, which in this first edition is devoted specifically to transposable elements. In the morning, we had a very wonderful session dedicated to transposable elements in eukaryotes and prokaryotes. Unfortunately, Dr. Doris Vela from the Pontificia Universidad Católica de Ecuador was not able to participate due to health issues, and Dr. Romain replaced her in that opportunity. And now, because of that, we are starting this last session with only three speakers. And this last session is devoted to tools and database for transposable elements detection and characterization. And we will have three great speakers: Alessandro Varani from the Universidade Estadual de São Paulo, Universidade Estadual do Estado de São Paulo in Brazil, Dr. Tyler Elliott from the University of Guelph in Canada, and Dr. Doga Eskier from the Dokuz Eylül University in Turkey. Now I would like to invite Dr. Simon Orozco Arias from the Universidad Autónoma de Manizales to moderate this session. Hi, Simon.
Hi, Vinicius. Thank you for the introduction. So, continue with our last session that Vinicius was telling about is about the tools and databases of transposable elements. We have our first speaker is Alessandro Varani. Dr. Alessandro Varani has a bachelor degree in biological science from the Centro Universitario Barão de Mauá and a PhD in biotechnology from the same University of São Paulo, both on Brazil. After that, Dr. Varani was a postdoctoral fellow at the CNRS in France and at the University of São Paulo. Currently, he is an academic at the Universidade Estadual Paulista in Brazil. His research is devoted to characterizing transposable elements through bioinformatics and system biology approaches, studying specifically the role of these in the evolution and genomic and metagenomic structure of microbial communities. Thank you, Dr. Alessandro, for being here, and please continue.
Thank you, Simon, and I would like to say thanks for all the organizers for inviting me to this incredible workshop. And I'm very glad to be here to present very recent results from our research group. It's a huge research group involving researchers from different places, where we developed a new database related to catalog prokaryotic transposable elements. And the name of this database is TnCentral, okay? And I would like to present this database, but before, it's very important to describe or remind all you guys here. We had a very nice presentation at the beginning of the morning by Professor Marianne, which she showed a little bit about these prokaryotic mobile genetic elements, and I would like to describe a little bit more about these elements. And the bacterial genomes are composed by a several number of genetic and mobile genetic elements, and these elements are known as genomic islands, prophages, conjugative transposable elements, or ICE, superintegrons, integrons, unit transposable elements, compound transposable elements, and insertion sequence, which are the smallest transposable elements found in bacterial genomes. One thing which is important to state is that all these elements can be mixed together in one bacterial genome, and this bacterial genome often carry plasmids, and these plasmids are plenty. Normally, there are plenty of this kind of mobile genetic elements. One concept very important that is the set of all these elements in one bacterial genome, we call it as mobilome. And the set of all these elements together with the plasmids, we call that as plasmidome. As you can see, we have different classes of bacterial elements here, and these classes can be grouped in different families, as we know in the insertion sequence and the transposable elements. In fact, we have a lot of different elements in the bacterial genomes. And as stated before, it's very clear that these elements are related with adaptation, emergence of virulence of different bacterial species out there, and clinical, veterinary, and environmental space. And you can see here an example of one plasmid, which are loaded of a number of different transposable elements. And the understanding of the mobulome assemble in these plasmids and in the bacterial chromosomes itself, it's very important. It's very important because understanding these elements in these chromosomes and plasmids, with the understanding of these elements, we can understand the evolution of these elements. Okay, and as you can see here, is a plasmid with different mobile genetic elements, and this is a result of manual annotation, and I'm going to explain it later when we will reach the TnCentral database. Okay, and it's well known that the transposase is one of the most abundant genes in nature. And this was described by Rami Aziz at 2010 in one nucleic acid research paper, where they used bioinformatic approaches, looking the public database, metagenomic database, in order to determine what is the most abundant gene, and they found that it's a transposase. We have, in fact, a multitude of transposable elements in bacteria, and these elements can be divided in major groups. We have the insertion sequence, insertion sequence-based elements, the TN3 family, TN7, TN402, and TN554 families of transposable elements in bacterial genomes. Look at the structure of these elements. What we have here in the upper, in the figure, we have one insertion sequence. This is the structure of one insertion sequence, which is composed by inverter repeats, as we can see here, IRR, and one transposase. And this element is flanked by direct repeats. We can say the same for eukaryotic started site duplication. It's a scar of the transposition mechanism. We also have a different class of IS elements, which is one IS which can carry a passenger gene, shown here in green. Okay, it's exactly the same thing as a insertion sequence, but with one passenger gene. We have the compound transposable elements, which is composed by two insertion sequences, as we can see here, two insertion sequences, and in between these two insertion sequences, we have passenger genes, some of them related with antibiotic resistance. Okay, and we have the transposable elements, the unique transposable elements in bacterial genomes, which are composed by the transposase and one resolvase, and also the passenger genes. These are the main structures, but we have a lot of exceptions of these structures derived from the structures. I like to say that biology is the science of exceptions, right? And we don't have an exact rule to describe all this diversity of transposable elements in bacterial genomes. And one exception is this one-ended composite transposable elements, which is formed by one insertion sequence in one site, and only the inverter repeat in another site, okay, as we can see here, and a group of passenger genes. We also have different structures, but this structure is from the insertion sequence family group, ISC1380. Okay, it's formed by a transposase, invert repeats, but we have also others invert repeats located downstream or upstream these elements. And these invert repeats can be form the transposition module and carry all other passenger genes which are located nearby this transposase. Okay, oops, sorry. And we have other derivatives. For example, we have the non-autonomous elements derivatives from the insertion sequence. For example, one insertion sequence which lost the transposase can form one mite. One mite element is a non-autonomous. And we have, as I said before, one insertion sequence which can carry passenger genes. It's also autonomous element. And we have another one which is very interesting, and I really like this one, which are the mobile cassette elements, the MIC elements, which are formed by only inverter repeats carrying passenger genes in the middle of these inverter repeats. Those elements are very underestimated in the genomes deposited in the public database because it's very hard to define, identify, and annotate this elements, and that's why important to do manual annotation of these elements, because only with manual annotation we can describe a better picture of the diversity of these elements in bacterial genomes. Regarding the non-composite transposable families, we have the TN3 family, TN7, TN402, and TN554, as I said before, and these families are formed, in fact, are composed by different structures, and I'm going to talk about a little bit about these structures. Starting for TN2 family, TN3 family is one of the first discovered, and this is the canonical element, the first one described, which is formed by a larger transposase, a TNPA gene, and a resolvase gene, and this transposase from TN3 is closely related to the insertion sequence transposase. It's a transposase with DDM catalytic site, and here we have the TNPR, the resolvase, and in between the transposase and the resolvase, we have a resolution site where the transposition, the co-integrate resolution occurs. And we have here a passive gene, it's an antibiotic resistant gene, of this flanked by short, small inverter repeats. Could be small, could be larger, depends of the member of this family. And the mechanism, the transposition mechanism, was elucidated decades ago, and it's forming co-integrates. And this is what happened. It's forming a co-integrate, and resolution of the co-integrate using the resolvase and the resolution site form the mechanism of this element from TN3 family. The TN3 family is very interesting because we have a multitude of different configurations of the resolution sites. For example, this is the classic element. We have the resolution site between the resolvase and the transposase, but we have other members from the TN3 family, like this TN5060, which is related with mercury resistance, where the resolution site is located in other position. You see, it's different. And you have other members with different resolution sites and different resolvases here. Okay. This means that this family is very diverse. This family is very diverse in terms of organization and passenger genes. Recently, we have found one new TN3 member which carries a toxin and antitoxin system inside the transposable element. And this toxin antitoxin gene, by bioinformatic analysis, we have found that probably this toxin system gene may be related with the transposition module and the transposition mechanism itself. This is just one example which we discovered 2-3 years ago. Okay, and the TN7, it's very interesting because it have a different transposition related genes, as you can see here, the TNS-A, TNS-B, which is the transposase. Okay, TNS-C, D, and E, and here we have other passenger genes, but in this case here we have one integron cassette inserted inside the TN7. And this integron cassette carries some antibiotic resistant genes here in red, as you can see, integrates here in blue. The TN402 is other example of family of transposable elements from prokaryotic genomes with a different configuration of transposable genes, as you can see here, and have a resolution site. The TN7 don't have a resolution site, and 402 has a resolution site located in the resolvase here, and also may carry integron cassettes carrying genes related to antibiotic resistance. And at last, not least, the TN554, it's a tyrosine site-specific recombinase, the TNPA. It's a different mechanism, it's a tyrosine, and can also carry important genes related with the resistance, bacterial resistance, and other genes related with virulence of bacterial genomes. In fact, when you see all of this diversity of different elements in bacterial genomes, this diversity and this complexity make us, in fact, a need for the development of a very curated database of these elements in prokaryotic genomics, because our main focus is to understand the bacterial evolution and understand the impact of these elements on the bacterial genomes. And thinking a good database for transposable elements, we need to think about in high quality annotation. This is very important. And the annotation must contain elements which have interest for researchers or clinical community. Links to evidence, which means that our annotated elements need to be linked to a kind of publication where the element was characterized, preferentially, respect that. Standardizer vocabularies, ontology, annotation procedures, and human reader, friendly human reader interface, and very accessible in terms of computer resources. Thinking in all these features for a good database, we developed the TnCentral database. The idea behind TnCentral is to gather all transposable elements available, annotate them with high quality, and create one entry page for each element with the maximum information possible as we can catalog for each element. In the same manner as we have today at ISFinder database. ISFinder database is a database for insertion sequence. And the idea behind TnCentral is to be a database for transposable elements using ISFinder as a good reference, because we need the annotation of ISs to understand the structure of a lot of different transposable elements. This is the web page of TnCentral database. Here we have the page of the TnCentral database, the search page of the TnCentral. Here we can put one transposable element name and start one search, or you can start one search looking for a specific family or type, or resolvase type. You can choose your queries here in the search to find what we have in the database, the transposable elements which we have in the database. We have now more than 400 elements already loaded on the database, manual curated. Here, just one part to show the Transposon entry page. As you can see, we have a graphical display of the elements, of each element. In this graphical display, we show each component present in these elements. And when I say component, I mean open reading frames, inverter repeats, resolution sites. All those sequences are gathered and are shown in the entry page. Included reference, and one information which I really like is this one, evidence of transposition. We try to look in the literature if you find something, some group which describe this element experimentally, and if this element have some experimentally evidence, we can put here that there is evidence of transposition. This is very important to be stated, because we have several different elements described, but for many of them, we just have the annotation of this element, but we don't have any kind of evidence telling us that these elements are active or not. And these are on the database. This is the entry of each transposable element. And how it works, in fact, how we annotate these elements. At the beginning, we have bioinformatics scripts. We developed different kinds of bioinformatics scripts which scan bacterial genomes in order to find genomic hotspots which may contain transposable elements. Okay, and with this region described by the scripts, we loaded this sequence on the snap gene here. Okay, the first step is doing automatic annotation, generate one GenBank file containing the transposable element, the region containing the transposable element, and this region we load on the SnapGene. SnapGene is a commercial software. There is a paid and one free version. We can use the free version with no problems to do the curation of the elements. It's very easy. And to use the SnapGene, and SnapGene is a very intuitive platform to do annotations. I really strongly recommend SnapGene as a good platform for annotation. And also loaded on SnapGene, these elements use curation guidelines. I'm going to tell a little bit about this curation guidelines. We have a huge curation guidelines guide on the TnCentral. Using this curation guidelines, we do the proper manual annotation, generate an enhanced GenBank file containing all the features, and this enhanced GenBank file is loaded on TnCentral and create the TN entry page with all the information. One thing which is very important, all those elements can be downloaded in FASTA file or GenBanked, the enhanced GenBank file. And when I say enhanced GenBank file, is a GenBank file containing all the features present in one transposable element. Here one example of the annotation process, the TN21, it's a multi-resistance transposable element. And here we can see a proper and high quality annotation of this element showing integron cassettes inserted in the middle of this TN21. As you can see here, the integron inserted in the middle of this purple here gene. And you can see also the tags used on the annotation. And those tags are something like that. You see, it's GenBank style tags, the same tags used in GenBank database. And we are using the same kind of tags, using the same vocabulary from GenBank, just the information, which is a little bit different because we are doing the annotation related, specifically related to those elements. This is the annotation tags. And we have a curation guidelines, a very extensive document showing all the guidelines and all the procedures necessary to do a proper and complete annotation of these elements. The idea behind is to provide a very accurate annotation of all transposable elements from prokaryotic genomes. And with this high quality annotation, it's possible, for instance, to study the evolution of some members of these transposable elements. For example, here we are looking for the evolution of one TN3 element from the group 2021. Okay, and it started from this element, the TN5060, with a mercury resistance transposable element. And doing the high quality annotation is in fact possible to understand the others variants derived from this element. As you can see here, this is another element. And this element would occur one integron cassette inserted in the middle of this gene here. And you can see here all the other variants which can occur. And we can see that this is a very giant history of different variants derived from only one element, showing different events of integron cassettes insertion and other transposable elements insertion, like insertion sequence inside these transposable elements. This is only possible doing manual annotation, high quality annotation. Doing this kind of annotation, we can understand the history of these elements. And at last, the TNpedia the TnCentral also have the TNpedia. TNpedia is a Transposon encyclopedia. The idea is to build a Transposon encyclopedia using Wikimedia interface, which is the same used for Wikipedia. That's the idea of TnPedia. And we have a lot of information about prokaryotic transposable elements. We say general information of prokaryotic elements. We have one section here with one chapter for all insertion sequence families. And that means that all you need to know about insertion sequence is already here. And we are building the prokaryotic transposon families also chapters. We have some done. The TN402, TN554 is already done. We are doing the others. We expect to release this by next year. And what we have in TnPedia, for example, we have all the information for transposon, in fact, for insertion sequence families. Here, the IS6 families, for example, where you can have all the information related of the transposition mechanisms and etc. We have it for all insertion sequence families. And we have it for, at least by now, 2 different transposable element families like the TN554. This is just one quick snapshot just to show to you guys what we have there. It's a lot of information. I invite you, for all those interested to understand, to learn about these elements, to go to TnPedia and look at after this workshop, if you like. And one thing which I really like is the historical documents. As you can see here, we are scanning all the documents from the past showing the first insights about the prokaryotic transposition mechanism. And this figure was made by Mick Chandler and Galas in during the 80s, just to show one model of the transposition of IS1. It's very interesting. This have a very historical importance. And we are also in process to scan all the letters from Esther Ledberg. Esther created the first centralized nomenclature system for insertion sequence. She created the IC1, 2, 3, 4, and so on classification. All of these letters, old letters, are in the house of Mick Chandler, and we are planning to scan all these letters and release by next year on TnPedia, like chapters. Here's the paper. It was published a couple of months ago, I think 3 or 4 months ago, I think. And these are the information where you can find more about TnCentral. The idea is to continue development of TnCentral. We have idea to develop a database like a 4-layered cake, the Moblom as a 4-layered cake, as I said in the beginning, using S-Finder as starting database, TnCentral, which we have now, and in the future develop the TnCentral for plasmids with the annotation of the entire plasmids and all the configuration and assemblage of these transposable elements in the plasmids. And then in the next version, one comparative genomics interface, one toolbox for comparative analysis. This is our idea for future developments. I'd like to thank all the team involved with the development of TnCentral. I'm just one member of this team, and I'm here just only presenting these results, but this was developed for all this team. I would like to thank you, in fact, all these members, and thank you all your attention in my presentation, and I will be happy to answer your questions. And if you like, you can drop me an email or drop a message here on the YouTube. Thank you a lot.
Okay, thank you, Alessandro. Very interesting presentation. We have some questions. The first one I put here in the chat was doing for the Dr. Gustavo Izaza. He said that with Tel can work, Alessandro. These databases could be exported to ontologies representation as GO.
Yeah, in fact, what kind of ontologies do you have here? We have ontologies related with the transposase. We have ontologies related with the resolvase genes. We have different resolvase genes with different domains in each resolvase. And we have the passenger genes. I think this is the most important one. And those passenger genes, we can use gene ontology classification as well. I think it will be a good idea to use these representations in ontologies using with the passenger genes. I don't know if I answered your question exactly, but if not, you can drop again your ask here on the YouTube.
Okay, thank you. We have another. This is for myself. Speaking in thousands of sequences of trapezoid elements, thousands or maybe hundreds of thousands sequences, what are the main aspects that we should be taking into account in the curation of the sequences?
Well, one of the first aspects is the importance of the element. Most of these elements carry antibiotic-resistant genes. And this is very important. We need to track these elements to understand how the transmission of these antibiotic resistance characteristics pass through different bacteria by conjugation, etc. And this is one of the main aspects that we need to take in account for the curation. Start with the most important ones, okay? I'm also working with plant pathogens, and recently we have found one element which carry one gene which is very important for plant pathogens interactions. That's other example of a good aspect to look at this. And in terms of curation, unfortunately, we have a lot of, in fact, we have a lot of good bioinformatic tools which are capable to catch different genomic regions which may carry transposable elements. But we don't have a specific bioinformatic software. And in fact, I think it's very difficult to develop one because I was working on this kind of development during the past years. We don't have a bioinformatics software which is capable to manual annotated correctly these entire structures. Because as you can see in the presentation, they are very diverse with different configurations, and it's very hard to develop one thing to do automatically everything.
Yes, that's true. We have another question from Roman Guyot. How to identify and annotate non-autonomous elements in bacteria if they lack coding genes?
Okay, this is a good question. We only annotated non-autonomous elements if you look for non-autonomous elements. That's the point. And if you want to look for non-autonomous element, you need to do a manual annotation. The first thing which you need to do is to pick up all the inverted repeats annotated, manual annotated, and map these inverted repeats the entire genome. And then, with this result of this mapping, look what we have in between different inverted repeats. This is not an easy task, okay? This is not easy, and that's why this kind of elements, for instance, the Mickey mobile cassettes, are very underestimated in bacterial genomes. We know very little about these elements. Can you hear me? I lost the connection with Simon. Hello?
Yes. Simon had an issue with his internet, just dropped me a line through his mobile. But I have another question here from Iago Blanco. Yes, just a minute, let me organize here the questions. This is what happened with online transmission, live online transmission. Yes, here we have the question from Thiago. Can a tool like TnCentral provide high reliability prediction when you have lots of transposable elements? And the second question by him, does it reveal reliability of alignment for each sequence?
Okay, the main idea of TnCentral is to provide a curated database of transposable elements. All those elements can be used for a blast search. Okay, and this is a good starting point to find out different transposable elements in a given sequence. Okay, but when you put a new sequence, you need to think about that it's a new element which needs a different annotation because probably your new element in your new sequence have a different structure, different components. That means that you need to do a manual operation after this first stage of blast. Okay. In summary, it's a good beginning. You can use TnCentral to try to find different TN's elements in one sequence, but you need to do a manual operation. Okay, and the second question is?
Does it reveal reliability of alignment for each sequence?
Yes, if you use in TnCentral the database of transposable elements formed by transposase, recombinase, it's a good starting point to alignments and to search different elements in other sequence. Consider that.
Oh, sorry.
No problem.
Okay. I have another one from Romain. Can TnCentral be adapted to store transposable elements for eukaryotes?
Well, the question is yes, can be, but as you can see for bacterial transposable elements, there's a lot of different exceptions and different structures, different families. It's too complex. And our idea is to maintain in prokaryotic world and work with bacterial and archaeal elements. But can be. But we need people to work with us with that.
Yes, exactly. We have another, I believe the last question here. Let me check because I don't have it in text. I have only my mobile. From Rafael Marini. Do all transpositions indeed need a RES site to resolve?
Where's the question?
I'm trying to receive it here by staff because I believe we are having a small delay. Let me write it here.
Well, from TN3 family, it's necessary, but in biological terms, we have other families which do not carry RES site. Okay, different transposable elements, unit transposable elements which don't have a RES site. But depends of the mechanism. Okay, for TN3, it's a classical structure necessary for the mobilization for most of TN3 elements from the TN3 family.
Great, Alessandro. Thank you very much for your talk. It was wonderful. And now we need to move forward for the next talk. For the next talk, I'd like to invite Dr. Tyler Elliott